Till now we have studied genetic terminologies, monohybrid cross and dihybrid cross. Today we'll study Mendel's laws of inheritance, backcross and test cross. But first we'll study Mendel's laws of inheritance. In all, there are 3 laws. That is, the first law, it is called as law of dominance. The second one is called as law of segregation, and the third one is called as law of independent assortment. The first 2 laws, that is law of dominance and law of segregation, these are based on monohybrid cross, whereas the third law, that is law of independent assortment, it is based on dihybrid cross. So first we'll see these 2 laws which are based on the monohybrid cross. So for monohybrid cross, we can choose any character, as we have seen that there are 7 characters that were studied by Mendel. So these are the 7 characters and these are the traits of the characters. And the most important thing that you have to remember is the trait is either dominant or it is a recessive trait. So first we'll start with monohybrid cross. So this is monohybrid cross, and the example that we have taken or chosen for monohybrid cross is color of seed. In pea plant, the color of seed is one of the character that was studied by Mendel, and this character, it has 2 traits, that is yellow color and green color. So one is yellow color and the other parent, it is plant with green color seed. So the most important thing is, for any cross, parents are required. One has to be male and the other has to be female. What is the phenotype of the parents? Male parent is having yellow color seeds, whereas female parent, it is having green color seed. These are the phenotypes. So what will be its genotype? The genotype will be capital Y, and the genotype for female parent will be small y, small y. Then the next step is, what will be their gametes? Now for that you have to draw an arrow like this, and then we'll get their gametes as capital Y for male parent and small y for female parent. Since both are homozygous, we'll get the similar gametes. Instead of writing twice, we have written only once. Same for the female parent also. Now once we get the gametes, the next step will be the process of fertilization. So this is the next step, which is called as the process of fertilization, where fusion of male gamete and female gamete takes place, and then we'll get the next generation, which is called as F1 generation. So this is the first half of schematic representation or diagrammatic representation of monohybrid cross. These were the parents. From the parents we got F1 generation. What is the genotype for F1 generation? The genotype is heterozygous. One is capital and the other one is small, whereas the parent's genotype is homozygous. So for this heterozygous genotype, what will be its phenotype? So previously also I have told you, out of 2 alleles, when one is capital, then it represents the dominant one. Out of 2 parents, which trait is dominant? Yellow is dominant and green is recessive. That means the F1 generation will show yellow color seeds. So from this it is clear that yellow color of seed has expressed itself in F1 generation. So this is our first law of dominance, which states that when two homozygous individuals, 1 and 2, are crossed with each other, the alleles that appear in F1 generation are called as dominant. Out of two parents, the allele which has expressed itself in F1 generation is called as dominant according to the first law, that is law of dominance. Now coming to the second law. The second law is called as law of segregation or law of separation. That is our second law, law of segregation. Now for this law, we'll need the second half of our schematic representation or diagrammatic representation of monohybrid cross. Now second half is obtained after selfing of F1 generation. What we have to do? We have to cross the F1 generation with F1 generation only. That will be called as selfing of F1 generation. So what will be the parents for the next generation and what will be its phenotype and genotype? Now the F1 generation will act as second parental generation. So we'll take it as this is one parent which is having yellow color seed, and this is another parent which is also having yellow color seed. These are crossed with each other. The basic difference between the first parental generation and second parental generation is, in first parental generation both the parents are different and both are homozygous, whereas in second parental generation both the parents are same, but in this case its genotype will be heterozygous. So what will be its genotype? Its genotype will be capital Y small y and capital Y small y. Now this will be the genotype for second parental generation. After this, the next step is its gamete formation. Next comes the formation of gametes. What will be the gametes? It will form two different types of gametes, and the gametes will be, one will be capital Y and the other will be small y. These are the gametes for one parent, whereas these will be the gametes for the second parent. Now at this stage comes our second law, which is called as law of segregation. What does it state? When hybrid form gametes, the alleles, they segregate or they separate from each other and enter in different gametes. And the gametes that are formed, they carry only one allele, which can either be dominant or it can be recessive, and they express purely. Hence, the second law is also called as law of purity of gametes. So for the first law, that is law of dominance, we have to draw the schematic representation up to first generation. Whereas for the second law, we need the second half, that is after selfing of F1 generation till the formation of gametes. So this is second law, and the second law, it states that when hybrid form gametes, the alleles segregate from each other and enter in different gametes. The gametes that are formed, they carry only one allele, that is either the allele will be dominant or it will be recessive, and since they express purely, the law is also called as law of purity of gametes. So this is about the first two laws. Now coming to the third law, that is law of independent assortment. Now this law, that is law of independent assortment, it is based on dihybrid cross. In an organism, many characters are present and each character is controlled by a pair of alleles. Does one pair of allele affects or influences the inheritance pattern of other pair of alleles? To find out the answer, Mendel performed dihybrid cross. Now this is a dihybrid cross. The basic difference between monohybrid and dihybrid cross is, for dihybrid cross, two characters are considered, whereas in monohybrid cross only one character is considered. So these are the characters that are used. The parents are axial and purple. This is one parent. The second parent, that is female parent, is terminal and white. Axial and terminal, this is the position of flower. Purple and white, these are the colors of flower. So what are the two characters that are chosen? Position of the flower and color of the flower. So these are the parents differing in two contrasting characters. These are the phenotypes. These are the genotypes. These are the gametes, and this is F1 generation. So after F1 generation, the next step is selfing of F1 generation. After selfing, this is F1 generation, it is crossed with F1 generation. These are the genotypes and these are the gametes. Now these gametes, these are placed in the checkerboard. So this is the checkerboard where 4 gametes of male parent and 4 gametes of female parent, they are crossed, and finally we get 4 different types of phenotypes, and the phenotypic ratio for dihybrid cross is 9:3:3:1. What are the 4 different types of phenotypes? Axial and purple, axial and white, terminal and purple, terminal and white. Mendel performed many dihybrid crosses with different combinations, and every time he got consistent pattern of result and the same ratio in F2 generation, that is 9:3:3:1. From this, Mendel established the third law of inheritance, that is law of independent assortment. The word assortment means distribution. So what does it states? The law of independent assortment states that when hybrid possessing 2 or more pair of contrasting factors forms gametes, the factors in each pair segregate independently of the other pair. That means their expression is independent of the other character. So this is all about the 3 laws. Now coming to back cross and test cross. So first we'll see back cross. What is a back cross? Back cross is a cross in which F1 generation, it is crossed with either dominant or recessive parent, that is called as back cross. Now if we take an example, suppose the example of color of seed. What will be the phenotype for this F1 generation? F1 generation will be, all the plants will have yellow color seed. First we will take a cross of dominant parent. So that means the dominant parent will be also having yellow color seed. So these are the phenotypes of F1 and dominant parent. What will be its genotypes? The genotype for F1 will be capital Y, small y. For dominant parent, it will be capital Y and capital Y. Now the next step is gamete formation. This is homozygous and this is heterozygous. The gametes for heterozygous will be different. That is, one will be capital Y and the other gamete will be small y. For this, it will be both same. So either we can write it once or we can write it twice. It will be capital Y and capital Y. Now what we have to do is, we have to place the gametes in checkerboard. So this is our checkerboard. Instead of writing male and female, on one side the gametes will be of F1 generation and on the other side the gametes will be of dominant parent. So what are the gametes for F1 now? It is capital and Y and small y. These are the gametes that we have written on one side. Now we have to write the gametes of dominant parents. So it will be capital Y and capital Y. Now we have to predict the phenotypes. So capital Y and capital Y, just write it next to each other. What will be its phenotype? The plant will have yellow color seed. For this, capital Y and small y. Again, even if one alphabet is capital, it represents dominant. And which color is dominant? Yellow color is dominant. What will be the next one? Capital Y crossed with capital Y. Capital Y and capital Y, the phenotype will be, the plants will have yellow color seed. Same thing for this also, capital Y and small y. Since one is capital, the plants will have yellow color seeds. So this shows that all the plants, they are having yellow color seeds. So this is an example of back cross in which F1 is crossed with its dominant parent. For back cross, we will not cross the F1 generation with recessive, because same thing we are going to study for the next cross, which is called as test cross. So what is test cross? A test cross is a cross in which F1 generation is crossed with recessive parent only. This is the basic difference between a test cross and a back cross. In back cross, F1 generation is crossed with dominant as well as recessive parent. Whereas in test cross, F1 generation is crossed with recessive parent only. So for test cross, we'll take the same example. The F1 generation, what was the phenotype? The seeds were yellow in color. The recessive parent was, what it was? What was the color? The recessive parent was green in color. So these are the phenotypes. Their genotypes will be, for yellow color, since it was F1 generation, it will be heterozygous. That is one capital Y and one small y. For recessive, that is green color, it will be small y and small y. These are the genotypes, these are the phenotypes, and this is a cross. Now the next step is gamete formation. Again, since it is heterozygous, the gametes will be different. One capital Y, one small y. For this it will be small y and small y. Now what we have to do? Same thing, we have to place them in the checkerboard. On one side it will be capital Y, small y. On the other side it will be small y and small y. What you have to write down here? One is F1 generation and one is recessive parent. So just extend this. Now capital Y and small y. What is the phenotype for this? The phenotype is all the plants or the plant will have yellow color seed. The next one, capital Y, small y, same. The plant will have yellow color seed. Now this is small y and this is small y. Both are small, which represents green color of seed. Again small y crossed with small y. The plants will have green color seeds. So in the checkerboard, two plants are having yellow color seeds while the other two plants they are having green color seeds. So 2:2. 2 are yellow and 2 are green. If we reduce it, it becomes 1:1. So this is the ratio for test cross. What is a test cross? F1 crossed with recessive parent, and the ratio for this that we got is 1:1. This is for monohybrid cross. If it is a dihybrid cross, the ratio becomes 1:1:1:1. 4 times 1. This is for monohybrid and this is for dihybrid. Why it is 4 times? Because in dihybrid cross we get 4 different types of genotypes. Whereas in monohybrid cross we get 2 genotypes, hence it is 1:1, and for dihybrid it is 4 times 1. Thank you.